So dear students, in our last lecture, we have discussed about the function of adjuvant and how adjuvant is boosting the immunogenicity of an antigen and why adjuvants are necessary for the preparation of vaccine. Today, we are going to discuss about two things. One is antigenicity and immunogenicity. Second one is the types of antigens. So first we will start with immunogenicity. Immunogenicity is the phenomena of an antigen, or we can say this is the ability of an antigen to induce both humoral as well as cell-mediated immunity. Means mostly they are inducing the acquired or adaptive immune response of the host cell. And antigenicity means it's the ability of an antigen to complex with an antibody and how it combined with an antibody specifically with the final product. And here the final product is nothing but antibody. So complexation between antigen and an antibody is known as antigenicity. So here we are going to discuss about the background of immunogenicity. First, the antigen presenting cell is the APC here. APC will actually go for processing and presentation of the antigen, and APCs plays a critical role in immunogenicity of an organism. This process is performed by immunogenicity is performed by antigen, and here the antigens are recognized first by the antigen presenting cells such as the dendritic cells, macrophages, monocytes. There are two involved events in this process. The first step, the antigen is captured by the APCs and then delivers to the cellular antigen processing machinery. These extracellular antigens are captured by the APCs, and the process of capturing maybe phagocytosis, maybe macropinocytosis, or maybe receptor-mediated endocytosis. So they are actually creating the endosome or the lysosome or the phagosome, and these are the acidic environment, the compartment of the cell. And under this acidic environment of endosomes or lysosome, antigens or the proteins are degraded into many immunogens, or we can say into many immunogenic peptides, which contains the T cell epitope. These immunogenic peptides contains T cell epitope. Here you can see T cell epitope is there. After acid hydrolysis within endosome or lysosome. Following the intake of antigens, the next step, MHC class 2, major histocompatibility class 2 molecule, is synthesized in the endoplasmic reticulum and then transported to Golgi apparatus to combine with the antigenic peptides to form a peptide major histocompatibility 2 complex. And in the second step, the antigen processing and presentation that generates antigenic peptides bound to the major histocompatibility complex molecules for presentation to adaptive immune cells. And these complexes are presented to the surface of the antigen presenting cells. Now, T cell receptor can recognize this peptide MHC2 complex and activate the T cells, mostly the CD4 plus T cells or the helper T cells. So the quality of the antigen presentation depends on the affinity of the peptide MHC complex, and there is a direct relationship between the peptide and MHC complex stability and the immunogenic responses. So this is the background of immunogenicity. This picture will tell you, here you can see how the degradation and maturation is going on. Here MHC class 1, this is a beta 2 chain of MHC class 1 heavy chain, and this is the transfer associated transporter associated with antigen processing, this cap. These are the transport proteins, the transporter associated with antigen processing, and this is the tap independent peptide form, and this is tap dependent peptide form. So in endosome, so first, if it is a receptor mediated endocytosis, the endosomes are formed, phagosomes or lysosomes. So then the antigens are cytosed, means this acid hydrolysis is going on. So via cross processing, they are getting unfolded and then interacting with the proteasome, or else they can be secretion. Here you can see that this nascent viral protein, this is the mature protein, they're getting denatured, and then these proteins are received by the MHC here, received by the MHC class 1 or class 2. Then they are transporting the protein to the T lymphocytes and inducing the T lymphocyte. And next, the T lymphocyte will release cytokines. The cytokines will induce the B cell, and now B cells either memorize it or else they will induce antibody production from the plasma cell. So this is the background of immunogenicity of an antigen. Now there are different determinants of antigenicity. I am discussing few here. The first one is molecular weight. First day when we are discussing about what sorts of proteins can be called as antigen. So then I have discussed that the proteins with a molecular weight 10 kd to 600 kd with complex structure can be considered as antigen. And here again I'm showing that this large molecule, very large molecule like hemocyanins, chitinous toxoid, having this molecular weight range are highly antigenic, so high antigenicity. There are some low molecular weight proteins like insulin. They still can show antigenicity, but their molecular weight is less than 5,000 Dalton or 5 kd. So they are showing low antigenicity. So I'm talking about proteins or complex proteins that can behave as an antigen, but there are many low molecular weight organic molecules that are non-antigenic by themselves. Eventually, they will not behave as an antigen, but they can show antigenicity, they can show antigenic behavior if they bound to a larger carrier molecule, mostly protein molecule. And when they combine with this protein molecule, this low molecular weight organic molecule now can show antigenic behavior. That is why these sorts of molecules are known as haptens. Here I have mentioned few compounds that can act as a hapten. So haptens can be defined as small molecules which could never induce an immune response unless coupled to a carrier molecule, that is a foreign protein. So what sorts of molecule can function as hapten? Dinitrophenyl group containing organic molecule, aminobenzene sulfonate, arsenate. These are the examples of hapten. Next is chemical nature. So almost proteins, they should be protein itself or polysaccharide or nucleoprotein or lipoprotein. So this is the chemical nature, means protein, this should be protein, either simple protein or else complex protein. There are some differences, some exceptions are also there. That all the proteins are not antigenic in nature. An example is gelatin. It's a high molecular protein, but still they are not antigenic in nature. So experimental studies have shown that compounds having aromatic radical is essential for antigenicity of a substance. Next property or determinant is foreignness. So degree of foreignness, how apart from the human. So an antigen must be a foreign substance, means of non-self origin to the animal, and that will induce more immune response rather than the self one. So antigenicity of a substance is related to its foreignness, or else we can say degree of foreignness. Next one is physical form. Again, experimental studies have shown that particulate antigens, means that are not soluble in water, are more immunogenic than the soluble antigen. Likewise, the denatured antigens are more immunogenic than their native form. Here in this figure, you have seen that these denatured one are more antigenic than their native form. So here, the native form, they are more antigenic than their native form. Now I'm coming about the types of antigen. There are 4 different types of antigen, broadly you can classify. One is exogenous antigen, means the foreign antigen, non-self antigen. Here the antigens are entering into the body from outside, and they are originating from a different host, a different source. And how they can enter into the host or the human? Via inhalation, via ingestion, or via injection. These are the 3 means through which they can enter into the human body. So their origin is different. They are entering from outside, they are foreign. That is why they are known as exogenous antigen. Next one is endogenous antigen, means this particular antigens, they are generated within previously normal cells as a result of normal cell metabolism, or because of virus or intracellular bacterial infection. The origin is self, means within the same host they are originating, not coming from outside. Third one is autoantigen. This is a type of endoantigen. Again, the normal protein or the complex proteins and some gene or RNA that is recognized by the immune system as an antigen, as a foreign particle. In those systems, immune system of patients suffering from specific autoimmune disease like rheumatoid arthritis. The fourth one is the tumor-specific antigen, tumor antigen, that can also be termed as neoantigen. And these particular antigens are presented by the MHC1, major histocompatibility, one or two molecules on the surface of the tumor cell. They can sometimes be presented by the tumor cell and never by the normal cell. So these sorts of antigen you will never found at the surface of the normal cell, but you can find it at the surface of the tumor cell. That is why they are called tumor-specific antigen or TSA. Next is a different one, superantigens. What are superantigens? Superantigens are also exoantigens. Mostly they are of virus or bacteria origin. So proteins produced by pathogenic bacteria or viruses. So they have the ability to stimulate a large proportion of T cells via interaction with the variable domain. Here you can see interaction with the variable domain of the beta chain of the T cell antigen receptor, TCR. Here, this is the helper T cell, CD4 plus T cell. Here you can see this is the variable part and the beta chain. This is the alpha chain. This is your T cell receptor. Here you can see T cell receptor. So they are interacting with the beta chain of the variable domain of TCR. And these antigens will not process by the antigen presenting cell. So next slide will tell you what is the basic difference between a conventional antigen and a superantigen. So here you can see when the antigen is cleaved by within the endosome, we have some epitope, and these epitopes are T cell specific epitope. So they will bind and form a complex by the help of the cell that is known as antigen presenting cell with the association of major histocompatibility complex 1. And this is the conventional antigen. Now they are processing and presenting by the antigen presenting cell with the association of MHC class 2. And here is the T cell, CD4 plus T cell that has a specific receptor, TCR. And this antigen has an epitope that can bind with this T cell receptor and forming a complex. But here if you will see, this antigen is coming from bacteria. They are the exotoxin that is produced by the bacteria will bind with the beta chain of the variable portion or variable region of the TCR. Not they are complexing with here, and they will not process by the antigen presenting cells. They're not processed by the antigen presenting cells. So bypassing the antigen presenting cell, so they are actually binding with the MHC2 complex and T cell receptor beta variable region beta chain. So bypassing the conventional antigen recognition and directly cross-linking with MHC2. So there is no function of antigen presenting cell. Hence they are termed as superantigen. So if you want to contrast with the conventional peptide antigen, you will see that conventional peptide antigen, which generally activates less than 0.01% T cell, whereas superantigens are activating the T cells nearly 2 to 30%. So superantigen mediated immune response, immune activation, is likely to be responsible for some of the characteristic symptoms associated with infection with superantigen producing microbes. So what are the antigen producing microbes? Mostly Staphylococcus, Streptococcus. These are the bacterium that are producing superantigens and the poisoning caused by this bacterium. So this is the list. You can see staphylococcal enterotoxins, staphylococcal toxic shock toxin, staphylococcal exfoliating toxin, and streptococcal pyrogenic exotoxins. These are the examples of superantigens. Now I'm coming into antigen presenting cells. It's a very important component of imaging response. The cells with the capacity to capture. So what are the cells? We can say that these are antigen presenting cells. So those sorts of cells that has the capability of recognizing, capturing, processing, and presenting antigenic peptide to the T cells are termed as antigen presenting cell. So they are presenting the antigen to the T cells. And here this presentation is in the context of MHC class 1 or class 2 and also deliver co-stimulatory signals to the T cells leading to proper activation. And only the antigen presenting cells can activate the naive T cell. So what are naive T cells? These are also termed as T helper 0 cells. So these are the T cells that have differentiated in bone marrow and successfully undergone the positive and negative processes in central selection in the thymus, where from they are originated. Both the T cells are originated, CD4 plus or CD8 plus T lymphocytes originating from the thymus. One is the helper T cell, one is the cytotoxic T cell. So now these are the naive form of helper T cells, that is the CD4 plus cell, and the cytotoxic T cells, those are the CD8 plus T cell. So antigen presenting cells, there are two types of antigen presenting cells. One is called the professional antigen presenting cells or APCs, and the second one is the atypical APCs. These are the mast cell, basophils, eosinophils, etc. But predominantly take part in immune system is the professional APCs, and these are dendritic cells, macrophages, B cells, etc. Monocytes are also there. So these are examples of professional and atypical antigen presenting cells. So the difference between the two, amateur and professional, can check here that the ability, antigen antibody complexation, this ability is high in case of the professional APCs rather than the amateur APCs. So how this is processing, how they are processing the antigen, what is the pathway of antigen presentation by the antigen presenting cells? So first, the pathogen or the antigen will recognized by the antigen presenting cells via pattern recognition receptors. Next, the antigens will be processed, and then the processed antigen will be presented to the T cell, helper T cell mostly. Because the processed antigens have some epitopes of TCR, so they will bind with the TCR with the help of MHC1 and APC complex. So APC surface, they have two type of receptor, MHC1 and MHC2. So MHC1 will bind with the CD8 cytotoxic T cells, and the other side they will bind with the helper T cell via MHC2. So APCs, they are actually bridging the two type of T cells, helper T cells as well as the cytotoxic T cells. CD4 plus T cells, they are the helper T cells. CD8 plus T cells, they are the cytotoxic T cells, the cytotoxic T lymphocyte. So antigen presenting cells are presenting the antigen with the help of MHC1 complex molecule and interacting with the cytotoxic T cell, and again they are interacting with the helper T cell via MHC2 complex. Now the helper T cell will induce the B cell and activate the B cell to release antibody from the plasma cells. There are two types of B cells. One is known as memory B cell that memorize all the things, all the steps and the patterns and the incidences also. And the second one is a plasma cell. These plasma cells, they are actually producing antibodies. So this is the mechanism or pathway. This is the pathway of antigen presentation performed by the APCs. Thank you.